So let's take a look at problem 3.12. So in Drosophila, gray body color is dominant to ebony body color, while long wings are dominant to vestigial wings. So let's go with, for gray body color and ebony, we'll go with E plus for gray. So we can have two E pluses, or E plus E, and these will both be gray. And then ebony is going to be two little e's. And we have long wings, which are going to be V plus, or just heterozygous V plus V. These are long. And vestigial wings, which are kind of, just little inefficient, or I don't really know, but wings that aren't really there, or little nubs of wings or something like that. So vestigial is going to have two little v's. Vestigial or vestigial. Okay, so the question is, assuming that the P1 individuals are homozygous, diagram the following crosses through the F2 generation and determine the genotypic and phenotypic ratios for each generation. So if we have our ratios memorized, it shouldn't be too hard. But let's take a look here. The first cross is gray and long cross to ebony and vestigial. So the genotype, and they said, they already said in the question said that they're homozygous. So gray is going to be E plus, long is going to be V plus, and now we're not looking at linkage here. So these genes, the ebony, the body color, and the wing type genes are on different chromosomes, or they're so far apart on the same chromosome that they're essentially unlinked, so we don't need to draw them like this, like we've been doing in chapter 5. So we're just going to write the genotypes like I have over here. So for ebony and vestigial, so two little e's, two little v's. Now, okay, this is the P1, and we're going to cross these. Only one type of gamete can be made here because they're homozygous, so all the gametes have an E plus and a V plus. Only one type of gamete can be made over here. All of them have an E and a little v. Little e, little v, no pluses. And when these combine during fertilization, we have a genotype that looks like this. So all the flies are going to have a gray body color and normal wings, or long wings, because they have a dominant allele for each of those traits. Now we can think back to Mendel's dihybrid crosses, and we know the ratios in the F2 generation are going to be 9 to 3 to 3 to 1, more crossing individuals that are heterozygous for these two genes. And we also know the genotypic ratios 1 to 1, 2 4 2, 1 2 1. But we could use the forked line method just to be absolutely sure we get the right answer. So for the forked line method, we're just going to break these genes into a way to we'll just consider them one at a time. So if we crossed E plus E flies to E plus E flies, well, we know this is this is just like Mendel's monohybrid cross in the F1 generation. Going to F2, we are going to have 1/4 flies that are homozygous for the wild type allele, and 2/4 of them are going to be E plus E, and 1/4 are going to have 2 little e's. And we can do the same thing for vestigial. So if we were just looking at long wings versus vestigial, so we would know 1/4 would be V plus V. 2/4 of the flies would, 1/4 would be V plus V plus, 2/4 would be V plus V, and 1/4 would be 2 little v's. You can go back and you can take a look at problem, let me see, I think it was 3.7, where we do the dihybrid cross for yellow and round traits and pea plants. And this is the same thing. We're just doing the same thing for Drosophila with body color and wing shape. We can do the same thing down. Okay, so then of these flies, 2/4 of them are heterozygous for the wild type E and recessive E allele, ebony alleles. So of those, 1/4 are going to be homozygous for the wild type V allele. 2/4 are going to be heterozygous. 1/4 are going to be homozygous recessive. Same thing for these. Okay, and we can multiply these, and we'll have then we can figure out our phenotypic and genotypic ratios. So 1 out of 16, E plus, V plus, V plus. 2 out of 16, 1 out of 16. I can just multiply all the numbers together first. And here's our 1, 2, 1, 2, 4, 2, 1, 2, 1 pattern. And these are all going to be homozygous for the wild type E, heterozygous, homozygous recessive. These are all going to be heterozygous. So there's a pattern here. You can multiply them across one at a time, but you can also make out a pattern and make it go a little faster. And these are all homozygous recessive for E. Okay, so we have our genotypic ratios, and what do we want here? Genotypic and phenotypic ratios for each generation. So we should see a 9 to 3 to 3 to 1. So here are the genotypic ratios, right? 1, 2, 1, 2, 4, 2, 1, 2, 1. And the phenotypic ratio should be 9 to 3 to 3 to 1. We should see 9 that have at least one wild type E allele and one wild type V allele. We should see 3 that are homozygous recessive for E and have at least one wild type V allele. So remember, when I put this line here, it means it could be either the wild type or the recessive allele. 3 of them are going to have at least one wild type E allele and 2 little v's, and then 1, remember these are all out of 16, 1 are going to have 2 little e's and 2 little v's, and all of these are out of 16. So 1 out of 16, 3 out of 16, 3 out of 16, and 9 out of 16. And we can, let's just do the 9s here. So 1 here for having both a wild type, at least one wild type E and one wild type V. We have 1 there and 2 there. We have 2 here, 4 here, and we have 1 here. Nope, we don't have any there. Sorry. I can erase that. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. And what about the 3 2 little e's and 1 wild type V? Right here. And what about the 3 1 wild type E and 2 little v's here and here, and then the last one, I'll use this purple color, 2 little e's and 2 little v's. Okay, so we did it, and I knew it would be 9:3 to 1 because I have that ratio memorized. But if you didn't, well, then you could just count these up and figure out the phenotypic ratio that way. Here we knew what the ratio was, and we went back and checked to make sure we accounted for 9 of those, 3 of these, 3 of these, and 1 of those. Or a better way to say that is 9 of these 3 phenotypes, 3 of these phenotypes, 3 of those phenotypes, and 1 of these phenotypes. So that was part A of the question, and this could be, a really long question if it wasn't for one little thing. So, and that is, there's some shortcuts here. So I want to, well, okay, let's look at part B. So part B of the question is to map out this cross right here. So the next cross is gray and vestigial cross to ebony and long. And gray and vestigial is going to be, remember the question said these were homozygous. Gray is going to be E+, vestigial is going to be 2 little v's. Ebony is going to be 2 little e's. Long is going to be 2 wild type V alleles, so V+, V+. So, and if we cross these, well, we know these flies over here can only make 1 type of gamete. These flies over here can only make 1 type of gamete. So mix these flies together, fertilization happens, and we have F1 flies that have 1 wild type E allele and 1 wild type V allele. So all of the F1 flies have this genotype and phenotype, gray body and long wings. So, okay, so this is part B, just the first part. And that's, it's important to notice something here, because otherwise you end up wasting a lot of time. So let's go back up to part A. Okay, so here's part A. And I'm just going to copy this right here. Copy, and let's paste this. So here's part B right there. Can I fit this? It's part A right here. And I'm going to highlight A with, I'm going to draw a square around the question A, P1 and F1 generation, and I will draw a blue square around the part B. So the crosses are almost, they're very similar, right? So look at the P1 generation. So here in cross A, we had gray and long crossed to ebony and vestigial. So we had both dominant traits in one fly and both recessive in the other. In B, we have the dominant trait with a recessive trait. So we have gray with vestigial in one fly, and again in the other fly we have a dominant trait, long, with a recessive trait, ebony. So we just kind of switch, one of the traits. So instead of gray and long, we have gray and vestigial. Instead of ebony and vestigial, we have ebony and long in the other cross. And so the P1 is a little different, and the gametes the flies make are a little different, but the F1 generation is the same. So the F1 flies are heterozygous for the E and V alleles here. So we have an E plus and an E and a V plus and a V in the F1 generation of the part B. And the same thing down here in the F1 generation, we have a E plus and an E and a V plus and a V. So the F2 generation is going to be identical in both cases. So this question is arranged in a way to hopefully get you to realize that the F2 is exactly the same. So the F1s in both these crosses are genetically identical, so the F2 phenotypic and genotypic ratios have to be identical between the two. So you don't have to do the whole cross there. So let's take a look at part C. Part C, there's another shortcut here that we should know for, if you're taking an exam, you should realize, see these shortcuts so you can finish the exam on time. So with part C here, we are looking at gray and long flies. These are homozygous, crossing to gray and vestigial flies. So, and they're homozygous, so that means we're going to put 2 E pluses over here, and long is going to be 2 V pluses. And for this fly, it's going to be 2 E pluses and 2 V pluses. No, 2 V's. So this is the P1 generation, and you can see that both of these flies are homozygous for the body color gene. They both have E plus alleles, 2 E plus alleles. So all of the progeny are going to have 2 E plus alleles in the F1 and the F2 generation. So we can just ignore that for now and just treat this as a cross between long flies and vestigial flies. And then this is just a monohybrid cross, and we know that, okay, all the gametes here are going to be V plus. All the gametes from this fly over here are going to be V, so the sperm and eggs mixed together. And we have all flies that are heterozygous for the V gene, and they all have long wings. And we self these, no, don't self them, but cross them to each other. These are flies, not pea plants, right? So cross them to each other, and we end up with our, you can do the Punnett Square if you want, but we already have this memorized, right? So 1/4 V plus V plus, 2/4 V plus V, 1/4 little v little v. And we could put the phenotypes here. So these are going to be long, these are going to be long, and these are going to be vestigial. So yeah, at this point we could go in and we could say, well, we already decided all of the flies in this, in the F1 and the F2 here, are going to have 2 E plus alleles. So we could put that in here now if we wanted to. So pretty easy. So long wings and gray body, and all of these, long and gray, vestigial and gray. So the shortcut here is to realize that, 2 E plus alleles in both flies means you can pretty much ignore it and treat the cross as a monohybrid cross, which is a lot easier to solve than a dihybrid cross.